Welcome to the PharmaSource podcast, recorded on stage at CDMO Live Europe 2026 in Rotterdam. This episode is sponsored by PharmaBlock, a fully integrated CRDMO providing research, development and manufacturing services from early discovery to commercial production.
So we've been talking a lot today about speed and efficiency on this stage in the CDMO space. So our next speaker is going to put some real numbers on that. So Tian, Director of Project Management from ProBio, is going to show us how their integrated platform is compressing timelines down to just 4 to 5 months. So please welcome Leon.
Hello everyone, thank you for joining today's presentation. Talking about the biologic CMC development, speed was always top priorities, but this speed sometimes or most of the time treated against with the quality or the regulatory confidence. However, through today's show or talks, I would like to show you that this trade-off is not necessary. We can deliver with the real case. We can show you that we can deliver the tox materials from the single clones with a high concentration formulation in 4 to 5 months. Start from last year, we received this request from client. They want to develop the bispecific antibody. Background with the monoclonal antibodies, bispecific antibody have more chance to form the product-related impurities or some mismatch, and also they're tighter and the stability is worse than the monoclonal antibodies. But at ProBio, we have this well-established platform for the bispecific antibody development. They can be asymmetric or asymmetric format. They could be the single chain or the multiple chains. Over 50% of our projects are those kind of challenge molecules. We already accumulated a lot of experience, hands-on experience working with these challenge molecules. With all those knowledge, we established a toolbox which can help us quickly identify the problem and the root cause, and then to avoid the same issue happen again. So this is the AI-driven toolbox to solving these issues. The second request to come from client is about the high concentration formulations. Their target is to develop the 120 mg per ml. Either protein with a higher concentration is more prone to form the aggregates. And also with the increase of the concentration, they have much easier to form the increase of the viscosity, causing them more trouble for their downstream process. And here lists why everybody wanted to go to high concentration. Here is the track record or the, according to the market, like since 1998, about 1/3 of FDA-approved drugs are high concentration. This is a high demanding market. And with the high concentration, you can creatively new administration route, like you can use the subcutaneous to develop drug, then the patient can do their delivery themselves at home. And also you can develop some drug for the eye or for some special organs. This is the expanded application of the drugs. At ProBio, we already have established a platform and delivered more than 20 different kind of high concentration formulation products. We can help them to deal with viscosity issues, aggregation issues, or the pH change drift issues. The third question from the client is that they want to get their tox materials in 5 months, and also they want to get them from the single clones. On the current market, like a lot of CDMO claim that they can deliver the tox materials in 4 months, but most of the case, they achieve this accelerated timeline with a mixed pool or mixed clones. However, you need to provide the comparability study to show that your tox materials is comparable to your final GMP materials. And our client wants to avoid this risk, so they want us to use a single clone, and then you don't need to demonstrate that the similarity between your tox materials from the GMP materials. To achieve this, we have this called Express CMC package, which can help deliver the tox materials from DNA in 4 months. We have a real design, the process, and also the new technique, the plasmid. They can help us to achieve this. However, the limit for this package is that they can only working with the low concentration formulation, like 30 to 50 mg per ml. How can we using this package for the high concentration formulation packages? We need another tool, which is called a program service. You can consider this as a high quality transient samples productions. With this, we're using the same wholesale, same plasmid system which are going to be used for the stable cell line generation to produce these samples. So the sample will be more representative than the trending samples, and you can use this sample for the formulation screening. So this can save us 1 to 2 months for the overall timeline. I think I list all the challenge or requests from client and provide some solutions for each of them, which make what look like a mission impossible from the very beginning now look like more feasible. And for the following slide, I'll show you how this case is running. So the first step is to generate the high quality samples with the program service. For the program service, we got a plasmid, and then we do the transfection. After 2 or 3 days recoveries, and we do the fed batch cultures. The culture can run in 14 days, and then until the viability is still maintained very good qualities. Here is the results. On the left, you can see the viability and also the cell density of the cells during the fed batch culture, and also you can see their final purities, and the titer is 800 mg per liters, and this sample is very good for the formulation studies. For the formulation study, this illustrates all the general workflow from ProBio. We start with the pH buffer screening, and then with the selected pH buffers, we're going to do the solubility study to test or to establish a correlation between the viscosity and the concentration of samples. And then we do the excipients and the surfactant screenings. For the high concentration formulation, we'll have a risk assessment group from the very beginning with the full formulation to test the high concentration performance or stability of the samples. And after the solubility study, we're going to also have the viscosity reducer screenings to screening different kind of viscosity reducers to help maintain the viscosity within acceptable range. And through this solubility study, we're going to evaluate whether we can go through a liquid form or to the lyo form. Here is our result for this case. After 3 rounds of screening, we finally got to the final formulation, which is in a haze buffer with the sucrose and the prolines to protect the products, and with the PS80 as the surfactants. This bar graph showing 40 degrees on 4 weeks. We have almost no change in concentration, slight change in their purities, SEC purity and the CE purities. The PS80 change is within 10%, so it's a very robust and stable formulations for this product. Besides this formulation, we also need to consider the overall process, because the formulation development for high concentration is not just the formulation alone. It also involves some technique or process for the downstream, especially the UFDF process, because you're going to need to concentrate the sample to very high concentration during this filtration step. The most common problem you may meet is the pressure drop limitation and the pH drift. For this case, we didn't have the pressure issue because we have very good viscosity reducers, but we do have the pH drift issues. As you can see here, our target pH is 6.5, but however, if you use the 6.5 to do the buffer exchange, the final pH drift to a 6.7 or even to a 6.8. This is because of the Donan effect when you do the buffer exchange. So compensate the pH difference, we're using a lower, we establish the correlations, and then using a lower pH buffer to do the buffer exchange, then can maintain our final pH is still within the range. After all those formulation development issues, we also have some challenge for the bispecific antibody development. The first issue is to avoid the formation of the homodimer or some mismatches. The protein we're working with is a bispecific, it's asymmetric. So we're cloning using these 2 vector systems. We're cloning this 2 chain of this bispecific antibody to 2 separate plasmid, and then we can optimize their ratios by changing their plasmid levels when we do the transfections. Here I can show how it works. This is the original plasmid, which overexpresses 2 chains on 1 plasmid, and you formulate a lot of homodimers and also monomers. But after optimize these ratios, we have very good 40% increase in the purity just by changing the ratios. This is a big relief for the downstream to remove those all impurities and also increase the overall titers. Besides this, we also deal with the aggregation to remove those. For this part, we screening different kind of resins, including the CEX, traditional CEX resins, and also CHT type 2 and MMC, different resins to remove those impurities or the aggregates. By changing the elution buffer and also adding an extra washing step, we were able to improve the purity from 79% to like a 97%. It's a big improvement for the purities and remove all those aggregates. And then we'll talk about the timeline because it's one of the requests from our client. One of the reasons we can achieve the faster timeline is because this new vector system we have. We call it the ProMax vector system. On this vector, we have titer enhancer elements, which can significantly improve the titer. On the bar graph, it's very tiny, but you can see that we have very good performance for different kind of molecules, regular monoclonal antibody, bispecific antibody, or some fusion proteins. Another element we call the steady hold, which can improve the stability of the expressed gene in the host cells. On the bar graph, we show we compare several hundreds different clones. After 6th generation cells passages, we compare with their original cells titers. Their variation is within 10%. So with this steady up element, we can pick the clone directly based on their performance in titer and quality, skip the stability study, which save us at least 2 months to pick up the clones. So we can use this clone for the toxic materials manufacturers. And then I can show you the overall timelines. So for the first 2 months, we generate the high-quality samples, which can be used for the formulation screening. We have 3 rounds of the formulation screening. And in parallel, we're going to do the stable cell line generation to get our premier cell bank, and also develop the upstream and downstream process. And then generate the GMP-grade cell bank for the GMP-grade manufacturers, and also conduct the formulation verification. In parallel, we do the analytical method development and the qualification. The toxic materials were generated at 4.5 months, and following with the GMP batch, and then the subject data and also the characterization study. So everything can be completed in 9 months. I think this illustrates that through all those work and challenge we have, we can deliver the toxic materials in 5 months for these bispecific antibodies with a high concentration and also from the single clone. If your molecule is monoclonal, and we can do it in 4 months. And then I want to just give an introduction about ProBio. We already have more than 70 NDA clearance globally, including FDA, EMA, and also Australia, Korean. And we can working with the discovery to the preclinical development, and then to the earlier stage manufacturer and commercial manufacturers. We can working with all different kind of molecules, antibodies, fusion proteins, conjugated projects, and also some GMP-grade materials productions. And we also help our partner to license out their project to big company, MNC companies, and during this time, we can support the due diligence, on-site inspection to support our documents and also deliverables. And thank you for joining the presentations.